Thanks for having me here. The one thing, I have to narrow your view now because I'm only talking more or less about CO1. I know the disappointment in every vertebrate person here and your beloved 12S, but just take everything I say and about progress and subtract 90%, and that's where you are. And that's not a good news story then in this case. So what I'm trying to do is connect progress and reference library building and why we should have them, especially for those of us, me included by all means, who look more from a community level and do meta-barcoding. And meta-barcoding from a bulk sample as much as meta-barcoding from an eDNA sample. In that case, or in that sense, it doesn't make any difference to me what the source of my material is. The questions are different ones. But in order to do that, we'll have to deal with a huge amount of unknown. And depending on the questions we're asking or the applications we're using, we might be able to reduce that amount of unknown simply because of what we have already in our databases. Well, the good news for the Canadians here, we've done a lot more work for this part of the world than for others. My apologies for the people down south of the border, but that's what it is. So obviously, there's this big enterprise called IBOL around for quite a few years. Started out with the barcode 500K thing, so trying to barcode 500,000 different species, accomplished quite a few years ago. And now off to the next journey called BIOSCAN, wanted to end up 2028 with about 2 million registered species. That would surpass what we've done for the last 35 years since Linnaean taxonomy was actually brought up. However, I'm saying that very carefully because those 2 million will be rather genetic entities that we can, for the most part, liken to species. We can call them bins or OTUs, but they're not necessarily come with the full complement of a name, probably never get one because one of the most endangered species on the planet are taxonomists. So we will have a problem with putting names on all of them. But it depends on your question whether you're interested. The thing is, we need the names, we need the museums, we need what Chris's people are doing, tying us back to the stories behind it, because if I only have a label, there's no life history parameter I can tie it to unless I take that particular thing and watch it for the next 20 years of my life. So when you do a meta-barcoding studies, like we do in our lab quite often, you come to the point where you have this huge amount of data and then you're trying to make sense of it. You want to put labels on it. You can cluster, you can skip clustering, use ASVs or ESVs, whatever you want to call them, and then just see what are they. Well, one approach, and it's more widely used than you probably would think, is you take everything in a reference library like bold. So Jeevan is going to be here tomorrow. If you grab him, ask him how many times they have to throttle people that use bold with a script automatically. So they have a huge meta-barcoding data set fresh from the Illumina and then run it against bold, and every other bold user is sitting there waiting. Because our servers are big, but not that big. So it's a huge database, and it comes with a few problems if you run it against something as global as that, but at the same time as incomplete as it is. These are impressive numbers. 17 million records, 13 million have barcodes. 345 species with names. If we take our OTU called bin, then we're close to 1 million. The problem is, as you probably take from the map, most of those come from Panama and Costa Rica. That's where we sampled most. So it's a heavily biased database, as complete as it might look like. And one of my students noticed that very quickly when she was doing a study in southern Ontario. All she wanted to do is catch frogs, get a poop sample of those frogs because we wanted to know the diet. And we wanted to find a way that's the most uninvasive possible, because normally dietary studies on amphibians are very often done flushing their stomach and other weird things they do to the animals. We thought, hey, let's catch a frog, keep them overnight, see if they poop, collect the poop sample, take a fecal sample, do a DNA study of it. She did that. quite successfully so, especially for the northern leopard frog, which you can see here. And then ran 2 MySeq runs. And then we looked at the data, and you look at your species list that comes out of it, and then you have all these wonderful entries. Whenever you have something that's not yet in the database from your location, it usually comes to the next one somewhere else. So we're looking at a butterfly that is, if I'm not mistaken, a European species, because I know it from my old home, Collias croceus, showing up in this, and that's obviously no match. The danger here is clear. Imagine that's going to be a herbivore, and that is actually one where the larvae feeds on crops. Somebody could cry wolf immediately and said, oh, we have a new invasive species in Canada. Turns out, no, that's not the case. What you have to do is go through your data with a very fine comb and weed out these cases where you just have a mismatch because your library is both incomplete and biased. So, cautionary tale. So there's the other thing that shows up here sometimes. We have the bins, so an OTU, but as most other OTUs, whatever you can, especially when they come out of clustering, those are dynamic things. An OTU is not a stable entity through time. So I give you a guarantee that the bin numbers I have right now in my long list for this study that's ending this year, in 2 years from now, some of those bins will have disappeared. Yes, you can find them. BOLD has delta view capabilities. You can go back in time. So it's almost like in the genomics communities where you usually gave a date of your release of the genome with it to say, at point X, red list of bins. So that's another problem. Well, we can be a bit more selective. We can have subsets of that list that are, number one, as complete as we can get them, heavily curated by people to make sure that misidentifications, which I haven't mentioned yet, are weeded out. Here's one example. The problem is, it costs a lot of money to come up with over 100,000 barcodes, reference barcodes, and it's intense in terms of work. About a dozen people worked for months, half a year, maybe longer, just to come up with that list. These are existing records on BOLD. We accumulated and went through with experts, with taxonomists, to see which ones are really what we want. You see it's about 75,000 bins. Is that a complete Canada library? No. It's terrestrial arthropods and as many vertebrates as we have. Vertebrates are sort of complete, not really. There's still a few things which we don't have for this country. For terrestrial vertebrates, we're doing very well. But nobody has touched anything else, let alone dig a hole in the ground and looked at the soil biodiversity. I'm very sorry, but that seems to be the one thing we all keep aside. The good thing is, having a reduced library, number one, fast computation. You don't have to wait for ages. You have a much cleaner output, much less noise. Your matching requirements can be more stringent. If you have a very general library, you usually tend to ramp up your percentage of identity. So you allow only 3% divergence between them. That's a lot. With a library like that, you can do 1%. And you get a very clean picture, like this one. Same story, our frog is happy. The OTU table shrank a little bit from 250 to 222 matches. But to be honest, I trust this one way more than the one I had before. We removed all the lists of non-members of that list. Yes, we're ignoring the fact that there might be things that are in the general list that we have not encountered for our curated library, but still it's there. You can build these lists and all things, and you see I'm always using a bold view because you can use the data set functions in that system to build a library. So you pull records from different projects, have a way of communicating with others and curating it. So it's a nice help desk. Plus, in combination with the checklist function of the data systems, you can build on that. This is a context-driven one. It's called Pest Species of Canada. I have to say crop pest species. That is more clear. We have other pest species, obviously. Thing is, It's again work intensive. Chris mentioned that if you want to have a national checklist, you have to get all these entities together under one roof and then negotiate what you agree on. Yes, we are in a very fortunate position. We don't do that for the most part. What we did for this library is went into all the literature out there, federal and provincial regulations, and looked at the registered species and pulled them all together. Consolidating was not my job, it was these two guys, Chris Ho and Mohammed Ashfaq. It's a lot of work, especially because provinces not necessarily agree on taxonomic names. I guess most of you have seen that or heard about it. So it's a bit of filtering work, a lot of things, and then you have your list. Great. And then you pull and check in bold, you do in a gap analysis, how many have been sequenced, reliably so. So you do some quality assessment of your records, and then you have a wonderful list you work with. And then we did one little study where we tried to apply it with a meta-barcoding approach. So we went into experimental fields not far away from here, where Guelph, the University of Guelph and the Ontario Ministry for Agriculture sort of do joint research in crop. What we did is we placed some Malaise traps, you see one of them here, in different crop types over the course of one season. What we wanted to do is check whether we can notice all these pest species if they're around. And the other thing is, we also wanted to check internally in the lab, how quick can this be done? To answer the second question first, about a week if you really want to do it fast. And that's an interesting answer to any kind of farming operations, where sometimes it's probably crucial to have an answer within a couple of days. Because right now, the approach to pest species is usually blanket spraying at fixed times of the year. And I can tell you from the results of that study, it doesn't help at all. We've collected 2 sites, one season. In total, over 200 of the registered pest species showed up at one point in the year. Blanket spraying didn't change anything. Are there differences between the crop types? Yes, to some extent, at least for the pests. You see in these MMBS plots. And between sites when you look at all the total richness of everything. So there's one very simple answer to the crop type differences for pests. They feel much better if you are in a more bushy, low height environment, like soybeans or alfalfa, rather than in a big desert created by wheat or corn. No surprise, there's a higher insect richness in something that is more protective. It's their little jungle, and the Canadians have made maybe questionable decisions in terms of increasing soybean cultivation, because it's a huge export product that brings in a lot of money. So when you go around here and travel around the region, you'll see more and more soybeans popping up and everything else. So, 2 use cases. There's a lot of work that goes before. So before you go out with your own study, if you really want to do it well, you either hope somebody has done the work for you, like we did for some cases, or you have to do it yourself. And you can do that even in lesser known territory if you have partners that are willing to sample and work with you. This is Ecuador, one of the new BIOSCAN sites. So IBOL has some funding to support Ecuador in building a reference library for terrestrial arthropods. So we started working there. Usually we start in low frequencies, like every 4 weeks we'll take a sample, or we collect more, but we pick only every second bottle or so from a lace trap and run it. And then you do a species accumulation curves to see how well does the sampling effort do. Well, and you see some of them were very quickly fairly close to saturation. That's not a surprise, because those are all from here, the Galapagos Islands. So, if you want to work in a low diversity environment, you want to do your reference library like this, go on an island. Don't go on the mainland. And it's nice because now we can do something that we've started doing repeatedly, and that's the only sort of DNA angle I can bring today. We're looking at bird nests of all things. This was an initial study to proof of concept with Val and Bettina to see if we can take a bird nest after use, filter the debris, the dust out of it, and do a DNA extraction from the dust. What type of DNA do we get? And there's a lot. As much so that you can start differentiating between, was this a prey item, is that a commensal, are those symbionts, or perhaps more importantly, are there parasites that have an effect on the nestlings? So that works quite well, and we thought, well, if we have a nice library in Galapagos, why not doing it there? Taking one of Darwin's main species, the Darwin finches, which are currently under huge threat by a botfly, a parasitic fly that kills them essentially, or at least leaves them with a deformed skull. What they do right now in order to fight this, one thing is to apply pesticides to the nests. Yeah, but it's the only thing. These are very small populations. An impact like that could wipe them out very quickly. Plus, there are textbook examples for evolution. So they do that, but they're also interested in finding out the blanket application of a pesticide, how much does it have an influence on the community that's probably beneficial. Now that we know a little more about who's there, we can take our nest samples, and they just be finished collecting them, sift through, take this eDNA approach, and find out who's there. Is the parasite there? We have control nests that have not been treated, so we can actually start doing comparisons. And one of my students is going to head out in a month down there to Galapagos and do that. So, a couple of examples why we need reference libraries, but we don't have to all run towards the large, big, all-encompassing ones. There's a purpose-driven one. My plea is put some effort into building it or help building it. Join forces. Talk to each other on these kinds of meetings. Say, can we do it? We don't have to reinvent it. I see with the fishes of Canada, a lot of repeated effort still. Multiple people doing the very same species over and over again, probably even for the very same marker that has been done already. More communication might happen. So, and that is my last slide, where I thank everyone in my lab at the center and our founders. Thank you.